Here's a preview of our next couple of topics here in Gen Bio. This video is going to mostly be concerned with how cells divide. Then I'm going to talk a little bit more about cell communication towards the end of this video. And from here, we'll start transitioning over to talking more about how cells transmit information from parent cell to daughter cell. But the main topic for this video is mitosis, also known as the dance of the chromosomes. So by now you've had a chance to see a short video clip that shows mitosis as it's happening in a blood lily. Here's also a really beautiful picture of midway through mitosis in a wide range of different types of cells. And so now we need to talk a little bit more about the details of this. We've also learned already about the M checkpoint, which is a point in the middle of mitosis where cells will pause to make sure that all the chromosomes are attached to spindles at the metaphase plate. And as I mentioned, we're going to start to understand what that sentence actually means. But before we do that, we need to do one other thing, which is, if we're going to be talking about the dance of the chromosomes, we need to talk about what a chromosome is. So to set the stage, when we've been looking at the nucleus of a cell, we've seen nuclei that contain a whole bunch of loose DNA. And when DNA is loose or unwound, it's referred to as chromatin. And DNA has to be loose in order for processes such as transcription and replication to take place, because the proteins that engage in transcription here and replication need access to individual strands of DNA. So if the DNA were wound up, then the proteins could not actually access the DNA. In contrast, a chromosome refers to tightly wound DNA. And there are a lot of levels of organization to this winding, because if DNA were completely unwound, it would, I don't remember what the exact metric is, but it stretches for an incredibly long distance, and yet all that DNA has to be packed into a single nucleus. So if we examine DNA on an initial level, there will be individual strands of DNA, right, composed of two anti-parallel strands. Those then get wrapped around a set of proteins called the core histone proteins. So that's the first layer of organization. And then along a stretch of DNA, there will be a series of histone proteins that the DNA is wound around. And then those interact with each other to form a more dense structure. So it looks like a nice knit sweater or something of that sort. There's supercoiling that then happens, and even more proteins added on that form a scaffold or a structure to keep everything organized. And so then eventually we reach an organizational level where you can actually see under a microscope in many cases the pieces of DNA as a chromosome. So when it's in this format, the DNA is really tightly wound, and so that helps with ensuring that DNA is not going to get tangled up or get lost in the process of cell division. So we need to talk a little bit more also about some of the anatomy of a chromosome. So this whole thing here is a chromosome, and we're going to talk a bit more about that in a moment. When it looks like this, this chromosome is composed of two pieces of DNA that are joined in the middle at a region that is referred to as the centromere, so a connection point. So we've got two pieces of DNA that are joined like this to each other. Okay? So these two pieces of DNA, there's two arms to it. So each arm is referred to as a sister chromatid. And so each of these sister chromatids is identical to the other sister chromatid that it is attached to. Okay, so two sister chromatids make up a chromosome, and each sister chromatid is its own piece of DNA. All right? So, having said that, let's now take a look at mitosis. So, there are 4 stages to mitosis. This is just a general overview. We're going to go through each of these stages in a bit more detail. And I'm actually going to add a stage and a half at one point. But there are 4 stages to mitosis. So there's prophase, metaphase, anaphase, and telophase. So 4 phases. And you can remember the order by just remembering PMAT. Easiest way to remember that. So as part of this process, what happens is there will be a set of microtubules that are going to anchor to those centromeres in the chromosome, and they're going to anchor at a protein structure called a kinetochore. So these microtubules are referred to as kinetochore microtubules. So breaking some of these words down, so kineto or kinesis refers to walking, and then microtubules are small tubules. Okay, so in earlier stages of mitosis, one of the major processes is those microtubules anchoring on kinetochores at the centromeres of the chromosomes. And then in the process of mitosis, chromosomes are snapped in 2. And so those 2 identical sister chromatids get separated, and they move to the opposite sides of a cell. And so the end result at the end of mitosis, and after the cell as a whole has gone through cytokinesis, is the production of 2 identical daughter cells. So each of those daughter cells gets 1 of those identical sister chromatids. So let's look at these phases in a bit more detail, and we're going to continue studying these during our time in lecture. So here's another cartoon of the cell cycle. So we've got interphase in green here on the outside. And just as a reminder, so we've got the gap 1 here, where a cell is going to be busy growing, and it might go to the G0 phase. We've got synthesis, so that's when DNA gets replicated. So we go from 1 sister chromatid to then having the possibility of 2, because DNA is replicated during synthesis. Then gap 2 is preparation for mitosis, and now we're here at the first part of the M phase at prophase. Okay, so during prophase in a cell, the nucleolus, so that's a very condensed region, that will disappear. And during prophase, so pro kind of before the main business, so at that stage, that's where the chromatin is going to start that supercoiling and condensing into chromosomes. So that at the same time that's happening inside of the nucleus, there's also a fun event happening just outside of the nucleus with the centrosomes, right? So centrosomes present in animal cells. So in an animal cell, the centrosome is the origin of microtubules, and in preparing for mitosis, the centrosome is actually replicated. And so there will be 2 little centrosomes. Initially, they're right next to each other, and during prophase, the centrosomes are going to start migrating to opposite sides of the cell, or opposite poles of the cell. And so they're going to start forming something called the mitotic spindle. So these microtubules that are sticking out from the centrosome are going to start lengthening. So that leads us to pro-metaphase. I told you there was an extra step thrown in. So at pro-metaphase, the nuclear membrane starts to break down. So the nuclear envelope is going to come into pieces. And at that point, those kinetochore microtubules can now start to invade the space in the nucleus and can start to attach to the kinetochores, right? So they're originating from these centrosomes. They're going to grow out, and they're going to grow into the nucleus and hook up to the chromosomes. And there's also another set of microtubules that comes off of these centrosomes that are called the polar microtubules. And those are microtubules that don't hook up to chromosomes, but instead they're going to push against the microtubules coming from the opposite centrosome. So they're going to help keep those centrosomes apart from each other, kind of maintain some structure. Okay? So then that brings us to the next phase, so metaphase. And I always like to think metaphase starts with M, like the word midline. And at metaphase, basically the actions of those microtubules is going to continue, and it's going to cause all of the chromosomes in the cell to line up along the metaphase plate. So the metaphase plate isn't an actual structure, it's just a space. So it's kind of an imaginary line here in the middle in between where those two new cells are going to form. And just a reminder, this was another of the cell checkpoints, right? So it might take this chromosome on the left a little bit longer to work to its place along the metaphase plate, and maybe this one got there earlier, but everyone's got to be lined up together before this process can proceed. So from there we go from metaphase to anaphase. So I like to think anaphase starts with A, or with ana, which means apart. So anaphase is where those two, where the chromosomes snap apart, and I actually even like to imagine a little snapping noise as they separate at the centromeres, and now the sister chromatids are going to move to the opposite ends of the cell. So each of these sister chromatids is a single piece of DNA, and when it has separated from its identical sister chromatid, it's also now referred to as a daughter chromosome. So it's now a chromosome that's made up of just one piece of DNA after it has split from its identical sister. We'll talk a little more about that in lecture. So now I'm going to kind of interrupt working through this process to ask a question. So scientists have been really curious about this question of how do these chromosomes actually move? And to make this point, I'm going to show you a quick little demonstration here on the desktop. All right, so I had to get kind of creative with the office supplies here to work on this demonstration. So here is my set of identical sister chromatids, right? And at metaphase, they're going to be joined here at a centromere. And then they've got these microtubules, kinetochore microtubules, attached at a point here at the kinetochore. So one of the things scientists have really wondered about is how do these actually move apart from each other, right? And there's two possibilities. So one possibility is if I take these pieces of string, right, it could be that they get pulled apart at the centrioles, right? They could get tugged along like so. So that's one possibility. Another possibility is that instead of getting pulled apart from out here, it could be that this chromosome walks along, and these bits here will wind up getting just kind of broken up and disintegrated. So it could be that it walks along this microtubule. And so remember, we've seen that kind of action before, right? Where vesicles use a similar process to walk along a microtubule. So, so how can we get to the bottom of that matter? So what scientists did is they figured out, well, what we could do is we could label a part of this string here, right, with a marker of some sort. And then if this chromosome, if the movement of this chromosome is a result of this pulling action, we'll be able to see the label move along with the chromosome. But if this chromosome is moving by walking along, then we're going to see that label disappear at a certain point. So let's take a look at this. So kind of rephrasing that, so they wanted to know how do chromosomes move. So here is the experiment that scientists have done. So they put a mark on those microtubule, kinetochore microtubules. They followed along, and hopefully you can see from the results right here which of those hypotheses wound up being supported. So as it turns out, the way these chromosomes move along the microtubule is they use similar little motor proteins, very much like the vesicles are using on microtubules, to walk along the microtubule to separate, which is kind of cool. Okay, so that's anaphase, and after that point the cells will enter telophase. And during telophase, things basically start to progress to a point where the chromosomes start to unwind into chromatin, and then the new nuclear membrane starts to form. And so this is kind of just the ending process. So we will no longer see chromosomes at telophase. And then the cell as a whole starts to divide. So the rest of the cell contents wind up being separated. And so a contractile ring forms between those cells. And if they're animal cells, that contractile ring will squeeze and it will create a cleavage furrow. And then you'll wind up with two identical daughter cells. So that's true for animal cells. If it is a plant cell, what happens instead are that, and you may have started to notice this in the video about the violet. So a bunch of vesicles start to form a new plate in the middle of the cell. And that plate then allows for the growth of cell wall and separation of the two daughter plant cells. So there isn't the same kind of squeezing action in a plant cell. And that's the process of mitosis. So now, for the rest of this video, I want to touch on one other topic briefly. So we're going to go back to thinking about cell signaling. So these checkpoints are internal signals for a cell that it's ready to progress to the next phase of the cell cycle. But obviously cells are also very sensitive to external signals as well. And so that it's a combination of internal and external factors that will jointly determine what happens with a cell. So cells depend on a couple different types of physical signals to decide what to do. So one of the types of physical signals is called anchorage dependence. So if a cell is just floating around and it's not attached to anything, it won't divide. It needs to be attached to something to say, okay, I'm somewhere where it's a good idea for me to divide. So it requires anchorage to proceed with cell division. So that's one type of signal that affects whether or not a cell is going to grow and divide. Another signal that cells rely on is density. So it's actually contact with their neighbors. And cells are also very much in contact with their immediate neighbors. And so if they sense that they have neighbors all around them, they will stop dividing. They'll stop growing. So this type of physical signal is referred to as density-dependent inhibition. And then on top of that, cells also respond to a whole bunch of chemical signals. So to give you one example, if you cut your arm, hopefully not too badly, and then need to regrow tissue at that location, cells that are at that location will release a molecule called platelet-derived growth factor. So it comes from the platelets that go to that site to heal that wound. And that platelet-derived growth factor will be a paracrine signal at that location to blood vessels to start growing and dividing. And this isn't just true for growth, it's also true for cell death. So there's a special term for cell death called apoptosis. And apoptosis refers to programmed cell death. So this is a little different from the cell death that we saw in response to a toxin. That cell death was not programmed, And you can imagine that if a cell wasn't planning to explode and it exploded, its contents are going to get released everywhere, and that could cause a lot of problems. So there are times when cells do need to die, but it's better if, when they need to die, if they die in a very systematic way, so they don't just spew their contents everywhere. So apoptosis is actually really important during development. So if it doesn't happen correctly, there can be a lot of developmental problems. And it can also be really important for proper immune function. So you can imagine if an immune cell did not undergo apoptosis, there could be big problems with what that immune cell might wind up releasing. So this kind of shows you some of the steps, and you don't need to know the steps of apoptosis right now. This is just more to illustrate kind of what it looks like, and to really drive home the point that this is a programmed process. So I'm going to wrap this up talking about kind of another sensitive topic, which is about cancer and the cell cycle. So most of the cells in the human body only divide somewhere between 20 to 50 times. And then after that point, the cells will stop, they'll age, and then they will die. So I'm really hoping that you're getting the message that the cell cycle control is a really intricate ballet. So it's really highly choreographed, and there's lots of signals and checkpoints and moving parts. So what that means is that there are a lot of different ways that things can go wrong with the cell cycle that will lead to the unusual behavior that we refer to as cancer. So cancer is effectively unregulated cell growth. So it's cells that are continuing to go through the cell cycle that don't know when to stop. They've stopped listening to the signals from other cells, and they're just continuing to grow and grow. And there are two main things that can happen when cells enter a stage where they're continually growing and dividing instead of stopping and listening to their surroundings. So in some cases, that cell growth might be localized, and those cases are referred to as being benign tumors, because those cells are going to keep dividing and dividing, and they might start to cause problems like, if that happens somewhere in your abdomen, where you have cells dividing and dividing and dividing and not stopping, eventually you might get a mass of cells that start to physically interfere with, organs. But you can usually treat that type of tumor by surgery and just remove it, and the person will be just fine. So that's one class of cancer cells. The other are cells that metastasize, so a metastatic or malignant tumor. And those types of tumors are the result of cells that don't stay clustered together, so cells that wind up traveling to other places in the body and start to colonize those other places. Just to give you one example, this is it's a difficult example for me to talk about a little bit. So my father died about a year ago from colon cancer. So he had a cancer that started in his intestinal cells. Those are cells that live in a really challenging environment. And so those cells started, or some of them mutated, they no longer responded to the right signals. And what often happens with a cancer like colon cancer is it might initially be localized to one place, but then over time it may start to spread to some of the neighboring tissues. So in my father's case, that spread to some of the lymph tissue that is very closely associated with the intestines. And once those cells start getting into the lymph system is a fluid system in your body. So those fluids wind up moving to other parts of your body as well. Then those cells started to travel to other organs. So, in his case, they wound up showing up in his liver, and then they started to show up in his lungs. And then we don't really know what happened from there, because past a certain point, you can't really pinpoint or localize where those cells have wound up. So my dad's case is a really sad case, but thankfully, in a lot of cases, there's a lot that we've learned how to do to treat cancer once we've identified that someone has cancer. And I'll just say, with the example of colon cancer, the real challenge in my dad's case was that it wasn't caught early enough, and that's very common to have happen with colon cancers in particular. And so I just want to say that it's really important to get screened for these cancers because the earlier that they're caught, the more likely it is that they can be caught at a stage where they can be treated locally, and then the person who's had that cancer has a higher chance of having a good quality of life through the rest of their life. But in any case, there's a range of ways that we treat cancers now, and they kind of vary in their level of precision. So one treatment method is to use radiation therapy. And what radiation therapy does is it just bombards usually a localized place with radiation. And the cancer cells tend to lose their ability to repair DNA. We'll talk more about that later in the course. So because they've lost that repair ability, they're much more susceptible to the damage and destruction from that high energy radiation. So they'll often be killed sooner than other cells in a person. Now, you're probably familiar with chemotherapy as another type of treatment, and the way that most chemotherapy drugs work is they basically interfere with specific steps in the cell cycle. So, they might cause cells to be arrested at a checkpoint, so they don't keep progressing through division. And because they interfere with steps in the cell cycle, so they disrupt cells from being able to divide, then when someone is on a chemotherapy drug, that's going to stop cell division for all of the cells in their body. So they'll discover that there are other cells like hair cells, cells in your digestive system that regularly divide, and so cell division gets shut down in those cells as well, and you see a lot of really severe side effects from chemotherapy, like hair loss. Immune cells don't divide, so someone on chemotherapy has a weakened immune system. So those are people that, in this day and age, have to be very careful about avoiding situations where they might get an infection. And just in the last 15 years or so, we've also seen some incredible advances in approaches for treating cancer. So one of those approaches actually uses some of what I've showed you with the immune system to help the immune system identify cancer cells and target them and take them out. So very similarly to the process of identifying antigens that I showed you earlier. Immunotherapy can often be much more specific to the cells that are causing a problem. But there's still fairly new therapies, so there's still a ways to go in figuring out how to make them work effectively, and they won't work effectively for all types of cancer. Along with that, we've seen a lot of advances in our DNA sequencing abilities, and what that has meant is our understanding of what cancer is has really changed a lot. So it used to be that we'd think of cancer as cancer of one organ. So I just mentioned colon cancer, liver cancer. With DNA sequencing, one of the things we've discovered is there's really no such thing as a single liver cancer. It's really liver cancers. And so knowing that means that we can identify, what has gone wrong in one of those specific cancers with a cell cycle, and that could provide us with opportunities to target those particular types of cancers more effectively. Okay, so that's how cells divide. We're going to keep practicing that process of mitosis, and then we're going to start to expand on it and take it in new directions in upcoming lectures.